each of us has the power to champion transformation. When you are confident enough to blaze new trails, because patient outcomes are so important. When you can tackle each challenge and invent new models to solve new problems. When you know that obstacles are inevitable but surmountable. With modern technology as your partner and AI and analytics as your guide. When you keep moving forward, then you'll go where everyone will want to follow. Conquering diseases together. Medidata.
Hello, everybody. Welcome to this talk. We are really very honored to have our guest. I want to say Glen de Vries because I am Dutch, but I think I should say Glen de Vries. That's probably better. Yeah, my name is Vanessa Evers. I am the director of NIST, the Institute of Science, Technology and Humanity. And we are really very honored to host this talk of Glen de Vries, who is the founder and president of Medidata Solutions. This is a huge cloud platform for life sciences, which you guys probably already know. That's why you came to this talk and why you are interested. What might be interesting for you to know is that this company has just been purchased by Dassault Systems for a whopping $5.8 billion. I can't even imagine what that is. If I translate that into like a stack of bicycles, I can't imagine how big the stack of bicycles would be. It's a huge amount. So congratulations to your company for that. So I think what is very interesting about Glen de Vries is that even though he's such a successful entrepreneur and is really changing the world of life sciences and medical sciences, is that he still feels at heart a student. Every day when he wakes up, he sees a student in the mirror and he has the interests and curiosity of a student. So I think you will find that you may have a lot in common. There's a lot to learn today. Please feel free to ask many questions. We have a system. Rachel, what is the system again? Slido.com. Please ask your questions. Feel free to ask any question. It might be that you would like to use this opportunity also to just ask questions about Glen de Vries' career, how he makes decisions related to the company, related to the talk. Feel free to use this opportunity to connect with him. Okay, thank you very much. Over to you.
Thank you. Good afternoon, everybody. Thank you for taking the time to come here and speak this afternoon. I will try to give you a little bit of my perspective on the world of life sciences, on the world of developing new therapies, new medical devices, digital therapies, maybe something that some of you aspire to do in your careers. Hopefully something that has at least some lessons and information relevant to whatever it is that you do in life. This is not a presentation about Medidata Solutions. Everybody who understands statistics in the room will know that coincidence is not always the same as causality, but I am the founder and president of Medidata, so I will talk about a lot of things that Medidata does, so it's at least coincidence. So, I do still think of myself as a student. I still think of myself as a biologist, and this is not a picture of my lab bench. This is a picture of a lab bench that I found on Google. But it looks exactly like what my lab bench looked like now almost 30 years ago. So I was a young researcher, and I was looking at how we could do a better job of diagnosing patients with prostate cancer and figuring out how to treat them. So I was using a molecular assay. I won't go through all the details. Suffice to say, it was an interesting idea, and I was publishing quite a bit based on this work. Now, for those of you who have done publications, who has actually published something in a journal yet in their career? for the students, was your name early in the list or late in the list of people on the publication? Early? Yeah, because the people who do the work are early in the list, and the people who get the credit are late in the list. And the person who was last in the list in all my publications was the most famous urologic surgeon in the world at the time. So not only did I have this cluttered lab bench full of all of the reagents and experiments that I was working on, but behind my lab bench was a freezer full of tubes of human blood that had been sent by every university in the world, basically, that was doing any kind of urologic oncology, any kind of prostate cancer research, because they all wanted to collaborate with us. And I was the one stuck trying to deal with tracking all of the blood, running all of the experiments, getting all of the data about these patients, integrating it and doing analyses and publishing them. And I was always somebody who was interested in computers and computer science. In fact, I went to Carnegie Mellon as an undergraduate, and one of my motivations was so I could do some computer science in addition to biology. So as a curious computer, maybe not professional, maybe not a proper engineer, but something beyond a hobbyist, I thought maybe I should create some kind of program that I could use to manage all of this research data. And that idea, which came from about 25 years ago, kind of was incubated in my head and the head of my benchmate, who, if I was on the scientific side of the university, he was on the medical side as a resident. And we kind of thought about it, and we started playing with the idea, and 5 years later, it actually got to the point that we wound up leaving the university. We left science, we left medicine, and we started what today is Medidata Solutions. Now, I am not putting up these numbers to try to impress you. I feel like I still have a lot of work to do in my life, but in the last 20 years since I started Medidata, I have accumulated a pretty big sample size for some of the things that I'm about to tell you about. So I have now worked with over 1,300 different life sciences companies, pharmaceutical companies, biotech companies, medical device companies, everywhere in the world. I have, to some degree, in some cases a large degree, in some cases just by association with the 3,000 Medidata employees that we have now, had some role in over 17,000 clinical trials. Over 17,000 instances where somebody said, I have a new drug, I have a new device, and it is safer, more effective, or more valuable for a patient than what exists today. Let me prove it. And so what I have seen in that time is a lot of things that are pretty old in the way they were executed from a scientific perspective. I don't know if anybody is a fan of Neil deGrasse Tyson. He's an astrophysicist based in New York City, which is where I'm based. And I'm a big fan of his. And I like that he talks about the scientific method as maybe not the perfect way to prove something, but it's the best way humans have ever come up with. Right? So most of the experiments that people run to show that things are safe and effective use a pretty simple scientific method that everybody knows about. In fact, the first clinical trial that's widely talked about was done in the British Navy, looking at sailors and what happens when you give some of them citrus, some of them lime juice, and some of them not, and who develops scurvy. And so you take one group of people, you give them the drug, you take one group of people and don't. And that's really how all clinical trials are run today. And you have this one-to-one ratio of patients who get your new drug, get your new device, and patients who get the standard of care. So can't we do something better? And this really is the thesis for a lot of the things that we do at Medidata, as I said before. Shouldn't we be able to, because we live in this digital connected world, look at data in new ways, connect with patients in new ways, and do something that is still has fealty to all the important methodologies that we use for science every day, but is more productive? Because remember, when I talk about this one-to-one ratio of the experimental subjects to the controls, I am not talking about something that is theoretical or something that I can go buy in a store, or just to be candid, even something like an animal that is not a Homo sapiens. I'm talking about people in our species right, who are volunteering to be the subjects in these clinical trials. So shouldn't we be able to do something better? And the answers that we've come up with over the years are related to some things around data science that I think are incredibly important, and I think they're important regardless of what field you're in. One thing that we've done very specifically at Medidata is we have tried to combine people from different disciplines. If you're doing experiments on people, you have to have people who understand medicine and who understand regulation and the responsibility, the ethical responsibilities that you have when you're doing those experiments. But if you only have people who grew up doing clinical trials, you'll have blinders on that will probably stop you from seeing new ideas. And so, as an example of bringing people together to think collaboratively, what I'm going to show you now doesn't come from my head. It comes from the head of our head of data sciences, who actually comes from the insurance industry. He put together the first predictive modeling group at AIG, the big insurance company. And one of the things that I learned from him is as you start to amass data, a lot of people think that, oh, well, the more data I have, the more valuable that is, right? And if I just put some steady effort into analyzing my bigger and bigger sets of data, I'll get incremental value, and it looks like a linear relationship. And what I learned from David Lee, the gentleman I'm talking about, is that that is completely incorrect. In fact, there is a curve that you have to climb as you put more and more effort into large data sets that most people don't have the time or the money or the gumption to climb. Because the beginning of it sounds boring. The idea of taking a big set of data and cleaning it and standardizing it and making sure that you have it semantically mapped to things that allow you to look at it in new ways is just not looked at as a sexy thing to do. And in fact, if you want to benchmark things, if you want to create interesting predictive models, if you want to do the kinds of things that I'm going to tell you about, you have to go through this effort. And probably a topic, you can ask me questions about it in a moment, but a topic not necessarily for today, but more of a business topic, one that I've thought about a lot over the last 20 years, is how do you create a business that actually has a model that allows you to continuously invest in this part of this curve, which normally people don't have that appetite or that budget to handle. But if you can start to climb it, you can start to do some really interesting things. So I said to you before, right, we look at a 1-to-1 ratio of people who are getting our new drug or device and people who are in our control group. Well, imagine each one of these circles is a different clinical trial. It's a different experiment done by maybe a different company, maybe for different drugs. All these patients are individuals, and everybody deals with a denominator in their statistics that is the size of this particular bubble representing their study. So as we started to standardize data, we realized that maybe we could start to get rid of those bubbles. Maybe if I'm developing a new drug, I used to work in prostate cancer, so I'll just use that as an example. Maybe if I'm developing a drug in prostate cancer, instead of trying to just find new patients to volunteer for getting my new drug or taking the current standard of care, maybe I can find the studies that people have done in prostate cancer before. Maybe some of those studies were done on the Medidata platform. Now, we do about half the research that's being done in the world, so you have a coin toss chance of that happening. So if we can find those studies, and remember we had a 1-to-1 ratio of people getting the new exciting drug to people who are just taking standard of care. Standard of care, simply put, means when if you walk into a hospital anywhere in the world, what are you on average going to get that's available today? Well, there's a lot of patients in those clinical trials who already got standard of care. So what we did is we put the effort into standardizing all of that data, into cleaning what were separate sets of data in different studies, so we could look at that as one single data set, and allowed our clients, to then do a new clinical trial, take this new circle, and actually in that same data model, compare the new prospective subjects, the new people getting this great new drug that should have all kinds of benefits that are better than what's on the market, to all these patients who had been tested before. And in fact, if you do that, in a statistically, mathematically, scientifically responsible way, you can actually, I don't know how many people have created one of these Kaplan-Meier curves in their career yet. I used to create them all the time. What you're looking at is a line representing the patients in terms of their survival, right? Survival might be something unfortunate like their death, or something maybe not quite as unfortunate, but usually it's associated with something unfortunate, like the fact that the tumor that you were trying to shrink has now begun to grow again. And here the bottom line is showing you what happens if you take standard of care. And the top line is showing what, in this made-up example, is the patients getting this new treatment. And you know what? This looks great. It looks like people who are getting my new therapy survive more than patients who are getting the standard of care. And so this is a great result. And what we call this, and by the way, we didn't call it this. Somebody found a publication about this. The earliest one we could see was from 1976. I'm reasonably confident that the vast majority people in this room were not born by 1976. I was. But the idea is old, but people weren't able to amass enough data and clean it to do this kind of thing at scale, what I was talking about from a business model perspective, until we came along and we were able to synthesize all these patients from other studies into a control group for a new study. I still think about myself as a student. I still think of myself as a scientist. So as much as time as we spend at Medidata making money, that's part of our job, running a company. We spend doing things that are academic still. This is a poster that we put up at ASCO a couple of years ago. We spend time in front of regulators and with not-for-profit organizations presenting things like why you should be able to use something like this in a statistically responsible way so that a regulator could approve a new drug, so a doctor could decide to prescribe a new drug, just like as if you did it in that old prospective 1-to-1 ratio. So, pretty simple idea, not trivial to do, but the returns are huge because now I can stop asking patients if they would volunteer in this experiment to be what is, if the experiment is a worthy one, probably the least beneficial thing for them in terms of just taking that standard of care. I go from a ratio, if you can see the text, sorry, it's a little gray, of instead of that being 1-to-1, it's 1 to much less than 1 in terms of prospective controls, because I'm using controls from all the other experiments that have happened recently, and maybe I have a control size now that is 10 times or even 100 times the size of the controls I would have prospectively enrolled in this study. Make sense? So this is a great step in the right direction. But maybe we can do more. And you're probably guessing already that we're going to go all the way across the slide so you can start to think of your questions and time them. That's exactly what's going to happen. I'm going to go until we all get to the other side of the slide. So what comes after this idea of synthetic controls? Well, let me talk about precision medicine. If you haven't encountered the term, I assume you have. I think the most simple way to state it is let's make sure that we, as much as we can, give the right patient the right therapy at the right time. That could be an interventional therapy. They're sick and I need to make them better. It could be something that is more of a prevention than an intervention. I want to make sure that this patient doesn't have a heart attack. Whatever it is, how precise can we be in terms of making that decision? Now, that's an idea that most of you are growing up with as a given, or at least an obvious endpoint. That's not really the way life sciences used to work. And this is, again, more of a business topic than a scientific topic. The practicality was, if you were inventing a new drug, a new medical device, you wanted to have a nice big market. And so you would look for a huge swath of people that you could potentially make better. And what you see represented in the gray parts of this slide are what happens when you go for a big swath of people. You say, okay, I want to develop a drug for people who have hypertension, or maybe anybody who has lung cancer. And if you don't know the intimacies of what this slide means in terms of phase 1, phase 2, phase 3 research, these are all gates before you get to that second to last part of this graph, which is that your drug is approved. Your medical device, your drug is going to be available commercially, at least in one country somewhere around the world and hopefully everywhere around the world. And if you can get through all these gates, this last gray bar, you're very lucky because it's very hard. Human biology is very complicated. And you have less than a 1 in 10 chance of giving this great new idea that you had in the laboratory, getting it to the point that it is approved for use in people. That is a pretty bad percentage chance, I think. And this isn't Medidata's data, this is aggregated across the industry by some consulting group. What's really interesting is when you start to think about this idea of precision medicine. So how do I be more precise? Well, I start to measure more things about the patient than just, oh, they have lung cancer. Maybe I'm looking for a particular kind of lung cancer. Maybe I'm looking for patients not just with hypertension, but patients who have a particular attribute. Maybe they have a particular gene. And if I just start to subset my patients, not based on an aspect of their disease, but based on an aspect of them as an individual. Let's use the gene as an example. They have a particular variant of a particular gene. You actually take the percentage chance of succeeding in terms of getting through all these phase gates and go from 1 out of 10 to 1 out of 4, just by measuring one extra thing about those patients. That seems like a pretty worthy thing to do. And what we're talking about in this example is really the idea of finding biomarkers. Finding markers in those patients that you associate with your drug, you associate with your device, and you connect to what in our industry is called a label, and what in the real world is literally a label. It's the thing that's on the bottle or the thing that's in the package that says who should take the drug. So you put a biomarker on your label, you're probably gonna have a much higher chance of getting your drug approved. And by the way, this is not just a great idea on paper, it's a great idea in reality. This is a study that we did with a group called the Castleman Disease Collaborative Network. You probably have never heard of Castleman disease. I hope you haven't heard of Castleman disease. It's an inflammatory autoimmune disease. Your immune system winds up attacking your own organs. It leads to organ failure. And in most instances, this is a life-threatening condition. The good news is there's a particular drug on the market. It's called siltuximab. And if you're a patient with Castleman disease, you have a 1 in 5 chance that this drug will completely control your disease. Okay, but 4 out of those 5 patients don't get any benefit. And that was discovered in research where people were asked to be volunteers, and half of them got this new drug, and 1 out of 5 of that half, so 10% of the patients in the research actually got benefit. And nobody knew how to identify which patients would get benefit after that research was done. Right? But what we were able to do is retrospectively start to look at some proteomics analysis. So we looked at the protein profiles in these patients. We identified some biomarkers. And we were able to go from a 1 out of 5 chance, a 19% response rate, to identifying a set of patients reliably, mathematically, who actually had a 65% chance of benefiting from this drug. Okay, so this is what I mean by precision medicine. So that's great, right? Great. I mean, we should be high-fiving in terms of the value we're bringing to those patients. But precision medicine has another slightly less fortunate mathematical side. Because the more precise I'm being in identifying the patients who will benefit from a particular drug, the harder it is to find those patients. Right? Now I only have, remember I was talking about the business of making a new drug? Well, my market size, if I'm developing this drug, just went down from 100% of Castleman patients, not many of them to begin with, to 20% of Castleman patients who actually benefit from the drug, right? It's not that we made the drug 65% effective. We were able to, in 65% we had a 65% chance of identifying patients correctly who would benefit from it. So patients become more and more scarce the more precise you get. The ultimate precision medicine is personalized medicine. We create something, and there's lots of drugs that are being created like this now that is based on the patient themselves. Take blood cells from Glen, do something to them in a laboratory or in a manufacturing facility, and introduce something back into Glen. The market size for that manufactured drug in the end of that process is Glen, right? Out of the 8 billion people in the world, there's 1 person it works for. Obviously, you're going to try to create something that's reproducible. But this idea that the number of beneficiaries of your development gets smaller and smaller is really important. So how do we deal with this? Well, Thomas Bayes to the rescue. And I hope that many of you have taken statistics courses already. If not, spoiler alert, I'm going to tell you that you should. And you know, if I'm permitted to be very trivial about it, Thomas Bayes was the first person who said that you can figure out that when you flip a coin, it comes up heads 50% of the time by just flipping a coin a couple times. Right? Other people would have said, well, we're going to design an experiment. We're going to flip 100 coins. And we're going to tally all the data. And we can't tell you anything about coins until we've flipped it 100 times. And Thomas Bayes said, well, if we keep flipping it, like every time I see it that it's 50%, 50%, I get closer to this percentage that it comes up heads versus tails, and we can actually start to use that data statistically. The idea of incremental knowledge based on every patient or every subject in your experiment, every data point that you measure, is something that really gets, at least from a publication perspective, traceable back to this guy, Thomas Bayes, right? Even there's a whole school of statistics. There's Bayesian statistics versus frequentist statistics. If you ever heard that and didn't know Bayesian was Tom, it's Tom. So what happens in clinical research when you start to think about some Bayesian principles? I'm actually going to show you a different diagram of this. This was the version drawn by one of my mentors, who's this guy, Don Berry. And he was one of the first people to really pound his fist on a table and scream from the mountaintops that people should use Bayesian statistics in clinical trials. And this is what that study that he's quite famous for designing works like. So instead of seeing an experiment which is kind of linear, right? So again, frequentist model. In my lab notebook, I write, I'm going to flip a coin 100 times. And this is how I'm going to record the results, heads versus tails. And here's my data. Here's all my table of all my heads and tails. And then I have my conclusion that I write with my summary statistics at the end, 50-50. This study actually is adaptive in its design. So what you see at the top is new patients entering this trial. And you don't have to look at every box. In summary, what happens is these patients go in. In the case of this study, they were all breast cancer patients. By the way, the study is still ongoing. And patients enter the study, and we look at a bunch of their genes, and they then get randomized, the same way we would randomize a patient to the new drug versus the control, right, in a typical experiment. They get randomized to one of N new drugs. Could be 1, could be 7. I think at times it's been as high as 8 or 9. And then we look at their outcomes, and we add that to our table of data, and then some other patient walks in the door and gets randomized. This is why this is about to describe, just shown as a circle, and why this is a learning clinical trial. Remember when I said when the patient first came in, we looked at their genes, and then we randomized them? Well, we didn't randomize them with a regular coin. In this case, they got randomized with a biased coin. They got randomized where if there were patients, let's say it's me volunteering for this trial, if there were patients who had similar genes to mine and they volunteered for this trial and they benefited from drug 2, my coin would be biased towards drug 2. If there were patients like me who every time they took drug 5, they didn't respond to it, my coin would be biased against drug 5. And every time this circle's gone through by another patient, we get better at biasing that coin, so we get more precise in terms of giving every patient who walks in the door the right drug that will be most beneficial to them. And it's not just breast cancer, it's being done in AML, it's being done in glioblastoma. This is the future. Don't take my word for it. Janet Woodcock is probably the most important person in the world in terms of the approval of new drugs, and she has multiple times publicly stated that this kind of adaptive design is the future of how life sciences needs to work. The idea that we don't have to test every drug in isolation is related to this first column. It's also related to this second column. It's the same element of discipline around data that is related to being able to look not just at whether my drug compared to a big data set, a big denominator is beneficial, but what is differentially beneficial to different patients. So we are now not just synthetically looking at people who got standard of care, but we're comparing every subject, and I think this is what's going to happen in life sciences. Every subject who walks in the door will be compared to every subject who has been in a previously executed clinical trial. And in fact, I don't think that's just something that happens necessarily in research. If you look at the idea of precision medicine, that is really what we're talking about. That you as a patient, and we're all patients at some point in our lives, at least at some point, all patients should be something that you can compare with other patients who have previously existed and what their outcomes were, so you can make the best informed choice. Okay, I'm still not done. There's still, I think, more that we can do. So these patients who are not in research, they're different than the patients in research. So when I do a clinical trial, people are represented by the green bar. You're doing an experiment. You only know the patient for a short period of time. But the green bar is pretty deep. Data depth is my y-axis here. I know a lot about them. I measure everything about them. Patients who just walk into a hospital or who have a health record at their physician's office, that's different. Your doctor probably doesn't know as much about you if you're not in an experiment as if you are in a structured experiment. So that's why the blue bars, patients in what in our industry we call the real world. Every time I say that, I have to say, I hate that term. Makes it sound like the patients who volunteered to be in a clinical trial aren't real. Of course they're real. These are patients who are often very sick and volunteering, but real world meaning not in a experiment. So they're being treated in the context of a non-experimental environment. From a data perspective, they look different. Remember I was talking before about this idea of progression, right? So it could be that the patient is done with the clinical trial because their tumor got bigger, or we see something in the patient's electronic medical record. We work in life sciences, we work with people. The way this slide is represented, there's no life after the X's, you'll notice. So in this example, it is death. And this is all looked at as separate data today. But if we start to, and we are just at the baby steps of starting this, if we start to apply the same principles of matching patients and homogenizing data, climbing that curve that I talked about before, why can't we take the patients who are in clinical trials and figure out how to, in one data set, line them up against real world patients? And in this case, even the patients that you see in the first, second, third, and fifth row who didn't die in the clinical trial, well, patients like them died shortly after from a disease progression perspective. So this doesn't look like a great drug to me. But if when I match those patients to similar patients, I realize that there are patients in the clinical trial who, in this case, didn't progress, they didn't die, and they looked a lot like younger, healthier patients in the real world, I think I can make a mathematical argument that this drug is much more worthy in terms of being prescribed, being paid for. I want this drug more as a patient. So there's another set of data that we can use from a comparator perspective, and that's data that's not experimental in nature. And I think if you look at what's happening outside of the world of life sciences, and again, it's not a topic for today in terms of the ethics, the morality, but we are all in our lives in one way or another starting to be measured in ways that use us the same way I'm thinking about real world data in clinical trials. We're being measured in terms of what we buy on Instagram. We're being measured in terms of what Amazon shows us first and what we're gonna buy. I think this is that same kind of idea, but applied to what I think is a very worthy idea. Okay, but I still have one more slot on the slide. And this is the one that I'm most excited about. Anybody know what this is? I didn't when somebody was telling me about it. The person telling me is Jerry Lee. He works at a university in California now. He was on the Joe Biden's cancer moonshot back when it was part of the US government, which is when I met him. And he's a chemical engineer by training. He's, I think, one of the most exciting cancer researchers in the world. Remember when I said multidisciplinary is important? So Jerry is talking to me about steam tables, which is what this is. So if you're building a steam engine, and I don't know, you're in the 1800s or whenever people built steam engines, you needed to know from any given set of temperature and pressure, how much water was vapor versus liquid. And these were experimentally built. So somebody would be sitting in a room with one of those glass vacuum vials, and they'd be measuring things and writing down all the numbers. And I was like, Jerry, why are you talking to me about steam tables? I don't even know what you're talking about. And he said, you know, steam tables, you know, remember phase diagrams? And I'm like, oh yeah, of course, I know a phase diagram. Well, a phase diagram is just a visualization on top of a steam table. That literally is all it is. That's in fact how the first phase diagrams, thank you, Professor Jerry, were put together. Right, so phase diagrams tell you at any combination of temperature and pressure what's going on with H2O. Okay, forget the slide. I have some medical news for you guys. And I'm speaking to the younger people in the room. If you are in your 20s, maybe you're not in your mid-20s yet, but if you are, and hopefully you'll get there, I have bad news for you and I have good news for you. Do you want the bad news first? The bad news is that if I look in the brains of cadavers, of people your age, I will find beta amyloid plaques. Not many, but I'll start to see them. If you're not familiar with them, we could take a bunch of Alzheimer's researchers and put them here and amuse ourselves as they argue for hours about whether that is coincident or causal in regards to Alzheimer's disease, but it's definitely coincident. So you already, or you shortly will, in your brains, have evidence that is consummate with an eventual diagnosis of Alzheimer's disease. That's the bad news. The good news is that most of you will die from a heart condition or cancer first. And that is really important to think about. And I use that kind of morbid example to put an exclamation point on how important this is. So imagine a phase diagram for Alzheimer's disease. Wouldn't it be great to be able to look at that and figure out for individuals, are there people who, of course, we're all on this trajectory where we're going to have Alzheimer's? You may listen to my presentation today and think, well, Glen's crazy already. But at some point on a long enough timeline, I'm definitely going to get Alzheimer's disease. But if it's so far out, if my disease trajectory is so slow that what is, I hope, the more likely circumstance that I'm going to have a stroke or a heart attack or have some kind of cancer that I need to deal with earlier, then probably I'm in the don't treat category for Alzheimer's disease. I'm 47. I definitely have prostate cancer in my body. I mean, I don't have prostate cancer from a diagnosis perspective, but the same thing happens. Once you're in your late 40s, your 50s, most men get prostate cancer. Your immune system, right, by the way, it's great at two things. Everybody knows the two things, right? It's great at fighting infection, and it's great at fighting cancer. Not the kind of cancer that you get diagnosed with at a physician. By the time your cancer has figured out how to avoid your immune system, right, then obviously your immune system isn't doing its job. But you have cells that start to uncontrollably grow and get these migratory phenotypes all the time. Your immune system takes care of them. So a lot of cases, like my prostate cancer, hopefully it's don't treat. But if it does start to grow, or if people do exhibit signs of dementia, then you like liquid turning into vapor or sublimation, you phase transition into needing to be treated. And of course, we want to treat you as precisely as possible. So there's different on-market therapies, there's different experimental therapies, and what we want to do is find the best one. So we talked about genes before, right? In 1865, this guy, Gregor Mendel, who's really the parent of modern genomics, figured out how to predict how peas were going to look based on their genes. Amazing. I mean, this work has transformed the world. And also, ironically, in February, many years later, I was already working at a teaching hospital. Two publications simultaneously came out with the sequence of human genes. And just like we were going to be able to predict what peas were different shapes and different colors, we were going to be able to predict what people were going to get what diseases and cure them all. It is now 18 years later, and none of that has happened. I shouldn't say none of that has happened. We have clearly derived a huge amount of value from the sequencing of the human genome. There are diseases like cystic fibrosis, which come from a particular gene almost all the time. They're actually referred to as Mendelian diseases because of that dude, Gregor Mendel, right? In science and medicine, right? We always have to use words that nobody else will understand, but Mendelian disease is related to one gene. But we didn't actually cure everything. In fact, a couple of years ago, this publication came out around how when you start to mathematically analyze a lot of diseases, from a genetic perspective, it's impossible to get back to a set of genes that are something you can identify as necessarily causal. I think it was the first time I saw this idea of omnigenic, right? So it's just everything in your genetic makeup has an impact, but it's not something you can use as a predictive factor. So let's stay on this topic of genes. Not only are genes really complicated in terms of predicting diseases, in some cases in terms of not being useful at all, but actually genes are kind of boring because they don't change. I hope a lot of you are looking at this slide and saying, Glenn, you are being hyperbolic. That is not true. Things change in genes, tumors mutate. My immune system does all kinds of interesting stuff with genes and they change, but that's the exception. Basically, all the genes that I have today in almost every cell in my body are pretty much the same as what I was conceived with. So I can't use the changes in my genome from a gross perspective over time to predict my health. If you've ever seen the movie Gattaca, it's a great science fiction movie, but I can't really predict who's going to be a good astronaut based on their genes. I can predict who's going to be a good astronaut next week based on how good you are at math and engineering and piloting today, right? So what I need to look at is phenotype. And phenotype is seriously complicated. It's always generating more data. In fact, I like the analogy of the Rosetta Stone. If you don't know the Rosetta Stone, right? Again, the topic not for today. The people wrote all over these buildings in ancient Egypt. I know it was only a certain class of people, but ostensibly it was like the center of human civilization, and then society forgets how to read all that data. But we got it back because we were able to find 3 versions of the same story, right? In ancient Egyptian and Damitic script and ancient Greek. And there was a key. Well, one of the things that I think we need to be looking on as kind of the new Rosetta Stone in health is let's look at all the different versions of what defines a state of health or wellness. It's the phase diagram, but it's not temperature and pressure. It's every single one of these dimensions and probably ones that I've forgotten about and haven't put on this slide. And that is a mathematical problem with an incredibly high degree of dimensionality. And one of the things that I was asked to mention in my lecture, and then I'm basically ready for your questions, is to talk to you about what skills I think are necessary in terms of solving some of these future state healthcare problems. And this is a representation of data in clinical trials, 2 dimensions per graph. And it's just an illustration of what you can do. The red dots are, in some cases obviously, in some cases very much not obviously, outliers when you do a machine learning-based analysis of that clinical trial data. Well, I believe that, and I think everybody should believe that if you were to try to create that phase diagram experimentally in every possible dimension, that is both impractical and, even if it was practical, certainly not ethical. It really is these disciplines around statistics and machine learning, which are going to be the things that allow us to look at these multidimensional problems of those phase shifts in patients, of how we actually capitalize on this future state of medicine, and how we do practical research that allows us to define those state boundaries. So with that, I will take your questions. Thank you.
Glenn, thank you. This was really a wonderful talk.
Thank you.
Not only are you a gifted speaker, you gave us a lot to think about. And there are people here in the audience, I think, from quite different backgrounds. Are there people who are more focused on medical background? Who's more into medical? Not that many. Computer science? Other life sciences? Is there a large group I'm missing out on? Someone. Wait, have you got a surprising background? How about you guys? Social sciences?
Nice.
Anybody? More social science?
Right.
All right. You're not
This is
alone.
a perfect multidisciplinary group.
Okay, great. So we get a little bit of the backgrounds. I really love this analogy of the Rosetta Stone. It's the Rosetta Stone with really many dimensions. And it's really compelling. But there's also something, I don't want to say scary, but there like a dark side maybe to it. And there were a few questions around that. So maybe if we start with that. One of the questions is, what are your thoughts about the importance of making precision medicine available to the mass population who need it more? than to keep them at a premium price. Who was this? Does anybody? I know it's anonymous, but feel free to address this question a bit.
Yeah,
if
but
you're.
If you don't want to put your hand up.
It's okay if you don't want to.
It's
If you
okay.
do, we'd love to hear what your thought about it, because it seems like a relevant question.
Or maybe you'll think my answer is wrong, and then you can tell me why too. Debate is good. So I have learned over the years something interesting around capitalism. I can tell you that when I first started my company, I met this guy who wanted to be our first salesperson. And I remember meeting him and thinking, he sat on this couch in our office, and I thought, how is there enough coefficient of friction between him and his shiny suit that he doesn't slide off the sofa? And I was like, there's no way this used car salesman is going to sell our amazing research software. Well, actually, in 3 weeks, I'm going to his daughter's wedding 20 years later. What I learned from him is that actually we needed to be able to invest in what we were doing at Medidata to create the platform that we have now. I bring that up because the world of life sciences is not different. And it took me a while to get there, I'm telling you, but I have realized that we need this reward to create an environment where it's even possible to invest in these research projects. Giving people around the world an experimental therapy in a controlled environment to the point that we can actually look at the results and responsibly, ethically say, this is something that this patient should or shouldn't get, is a very complicated and expensive process. Yeah, there's companies like Medidata that are trying to make it better, faster, cheaper, but it's still going to be complicated. It's still going to be expensive. And so I think it's important for us to realize we need to create rewards, incentives, to create an environment where people will invest in these new therapies. I also, obviously, think that any patient who is in need of a particular therapy is somebody who should get that within certain limits, right? Because I do think if you look at healthcare problems around the world, like there's a huge amount of money that is spent, for example, in the last days of most people's lives. I remember being at a conference and a guy who I thought was incredibly smart from the National Health Service in the UK was in the midst of all these people like me talking about big data and giant Bayesian studies. He said, you know what, just one bit of information. Does everybody in the UK, this was his mission, I don't know where he is with it, he said, we should know of all UK citizens who wants to die at home. And if somebody wants to die at home, we will let them do that instead of bringing them into a hospital and trying to spend all this money on keeping them alive for 3 days more or 5 days more. And I think he brought up something that from a per bit basis probably is one of the most impactful global health ideas I've ever heard. So that's why I say within limits. But I think that we can have societally our cake and eat it too if we think about this problem the right way. If we think about things like what people's personal preferences are, if we think about creating incentive systems as people are beginning to do, where pharmaceutical companies get an extended exclusivity from a patent perspective, if they can figure out how their drugs apply to diseases that might not be the first one that they invested in, I think there's a way that we can start to make that care available, that care more affordable. I do think that's an area that is ripe for innovation, but I don't think we have to choose between those two. This is not a place where we have to choose between a capitalist view and a more socialist style view of how society can and should be run, I think.
It's really very interesting, of course, because, well, if you approach it from a numbers perspective, you get very clear data back that says you should do this. Like the phase diagram tells you really clearly you should do this, but being sick is an emotional process.
Yeah.
Or going through, so that makes it a very interesting problem for governments and for hospitals to solve. Do you feel that you are having impact on that, on how these decisions are being made, whether that data is being used in a different way? In the hospital system?
Yeah, I think we're just beginning to get there. Because I think the reality is that most patients don't go into an environment where there really is precision medicine, right? They might be lucky and wind up in a situation where there's a precise therapy available for them, but it's not like we put people into this multivariate model and look at what the phase shifts are around them. But that's certainly the world that I would like to live in as a patient, and certainly we believe investing in.
So I work in hereditary cancers and rare disease, so Castleman's up my alley. So one of the challenges now in the field of rare disease is that such approaches are coming up with potential multiple targets, right? And they all sound relatively okay from the science perspective. And we're all facing this issue as a community now because next week I'm going to one of these meetings where to be able to actually successfully do a trial in any one of just these strands of these drugs, it's going to take another 100 of these patients where in a disease that there are only 20 in the world or 10 in the world, and you're going to have 4 or 5 different potentials out of the big data that might be hits there. So we're getting to a problem now of not too few solutions. In some cases, we have too many solutions. And I'm interested in understanding how you mathematically help us with that problem.
Yeah, so I don't want you to think that I feel like we've done something to solve this problem. I do have an opinion about how at least one of the solutions to that could come about. By example, I was asked to give a presentation at a stroke conference, and I know nothing about cardiology. So I figured the best thing to do was get up in front of the audience and tell them I didn't know anything about cardiology. But if I was speaking to a bunch of oncologists, what I would have told them, this was me presenting to the cardiologists, is that if you're interested in cancer research and you're looking only at data about patients in cancer clinical trials and you're trying to build a mathematical model around cancer diagnosis, you're looking in the wrong place because everybody already has cancer. So the place that you should be looking is in cardiology research, where you're getting this exquisite data run-up to the point that somebody is getting their diagnosis. And hopefully some of you are going, oh, but the cardiologist, I think, went, oh, yeah. And that is what I talk to oncologists about a lot. Like, we should be looking in these large-scale cardiology trials to find the data that we need for the modeling of the diagnosis and detection of cancer, or for Castleman disease, where there's plenty of patients who have that if you look at a big enough set of data. So the supersizing of the idea around reusing patients across trials is reusing patients across therapeutic areas. And again, that is a one notch in climbing the ladder of solving that problem. But I certainly think if we tried to your point, isolate all of these indications and create these tiny studies, we're never going to get there. We need to figure out how to look at patients more holistically from a research perspective.
Okay. So there's an interesting question by Joe. I'm just going to catch him and then come back to you. I don't know if Joe wants to ask his own question. If not, the question is, has Medidata faced issues regarding the accuracy or robustness of your models due to the erroneous or ambiguous data labels?
So, okay, to be fair, that's really our job to help our clients not be in that position. So they're the ones doing the research. Is that your question?
Yeah. Hi, thanks for taking my question. The reason why I asked it was just when I'm doing research in machine learning, and I find that whenever you build a model for something, you always you call it AI, but somehow you cannot escape the human in the loop somehow. I guess you've already touched on the topic of what data you choose. So it also comes in how the data is labeled, whether you label it correctly. So I'm wondering how much of a challenge there is in medical settings.
So good data labeling, having good semantic definitions is a huge problem to solve. It's really, that thing I circled around climbing the curve of data utility is really about labeling, right? And labeling and data cleaning. And so one of the things that we've at least tried to help our clients do in terms of that is looking at a bigger denominator as they look at individual data items. I can tell you just by example, we were just talking about at lunch. If you plot kind of like temperature and pressure, if you plot height and weight across all the patients who have ever been in a Medidata study, you get a nice little cloud that kind of goes up on a particular linear trajectory, and then you get these two little extra like flower petals next to it. And those flower petals are mislabeled data points. It's when somebody used centimeters instead of inches and kilograms instead of pounds. And so you can actually use some of the machine learning to try to figure out if your data labeling is consistent. And so I think this is an area that has plenty of innovation opportunities, but at least I know other companies besides us as well. We're trying to look at ways to automate that labeling and cleaning in a way that just makes the data that much more reliable. But you're right, it's a huge thing to think about. Garbage in, garbage out, right?
And thanks for the presentation.
Thank you.
And I'm Dr. Nagasundaram. I'm working in computational biologist. I'm basically, well, after the completion of Human Genome Project, and till date, we are having huge set of genomic and variation data. So, do AI have the capability to segregate or catalog the data for precise drug discovery?
A huge set of what type of data? Sorry.
Genetic data.
Genetic
Genetic
data.
data.
Yeah, the variation data, mutation data, and the genetic sequence data.
Look, there is a huge amount, I think, of drug discovery that is being powered by the cataloging of genetic data. But the thing that I think we always have to keep in mind is, just by example, how relevant, for example, is your microbiome on top of that genetic data? And if there isn't an obvious answer that connects a particular disease to a particular genetic condition, there's probably data that may not have even been collected in the research sample that you've got. So there's a lot of health systems, there's a lot of countries that are, I think, in a very admirable way trying to create connected data sets now, where you should, for the same patient, be able to look at their kind of natural history, you can look at their genetics, you can look at the environment that they're in, and try to weigh all these additional factors. Because there is no rule that says the genetic factors are going to outweigh another otherwise phenotypic one, right? And so the integrated data will be, I hope, more productive than just the genetic data. That doesn't mean that you shouldn't be doing research based on the genetic data. There's great stuff in there.
Okay, we are slowly running out of time. So there are some really interesting questions here about CRISPR, for instance, how to ensure that trials remain blinded, etc. But I wanted to end with two maybe more personal questions about you. So one of them is, what is your background? What did you study? And how did you combine data science and life sciences? And what did you do right after university? I think people are trying to figure out how to get to where you are.
Yeah.
What are the steps?
Okay, so if you're looking at me and trying to figure out how to get to where I am, remember, I'm an N of 1, so don't make any conclusions based on what I tell you. I studied molecular biology as an undergraduate. I thought I was going to get a graduate degree in molecular biology as well. I wound up leaving academia and starting Medidata, and then I thought, well, I can't live life without a PhD after my name, so I decided to go to graduate school in computer science. That's when I went to Courant, and I quit that as well. So N of 1, deciding not to get 2 PhDs is not causal with success.
I'm not really sure how to translate this. Please finish your PhD.
Yeah.
No matter what you do, please finish it. Get the ticket after do what you want.
Finish it. I always feel guilty saying that at a university.
And kind of finally, with all the data you've seen, all the knowledge you've accumulated, do you have advice on how to stay healthy as a young person? For young people. For me, it's too late. I'm done. I can eat all the hamburgers I want.
We're doing okay.
But these guys.
So I actually am pretty obsessive about tracking myself, just because I've seen everything that's happened. I've seen the backside of the world of medicine, because I worked as a research scientist at a hospital, so I know a lot of doctors, and I know that they don't really know anything more than anybody else. I see the seedy underbelly. I shouldn't say that. That sounds negative. I see the negative side of clinical research, the number of clinical trials where things fail, where the hypotheses aren't proven out, and that's a whole nother topic in terms of how we avoid those situations or realize that those drugs or devices are good for other things. I do think that treating yourself like that phase diagram is the best thing possible. I met a guy, he's unfortunately passed away now. His name was Jack Whalen, and he was an engineer, and he got diagnosed with a blood cancer. And he would show up to his doctors with an Excel spreadsheet. And this was 15, 20 years ago when it first started. And he literally tracked all of his markers himself, because his doctors weren't doing that. And I think more people are thinking this way, but he was so proactive around tracking his disease progression. He knew he was failing certain clinical trials before his physicians did. And so he was very proactive about his health. You guys are lucky. You live in a world where it's probably easy to track your heart rate variability, your sleep patterns, how much exercise you're doing, the way you eat. And I think, again, the world I want to live in as a patient is the world where I don't have to think about that or do that stuff myself. But that's my advice, is have a good grip on your own health status and realize when it's changing. It's probably my with no qualifications, my best health advice.
Thank you. Really appreciate it. Thank you so much
for your talk.
It was wonderful.